Good morning, my dear listeners. Today I discuss about the biosynthesis of purine nucleotides. Purine, which is the one of the most important base out of two base, purine and pyrimidine, and purine is the most important in this group. Adenine and guanine, which is the base for synthesis of DNA and RNA. This purine nucleotide basically it consists of the one 5-member rings and another 6-member ring, and this numbering is the 1 2 3 4 anticlockwise up to 9, and this N1 which is coming from the aspartic amino acid, and C3 is coming from the carbonate, glycine, N and formate. So purine nucleus is very important for the biosynthesis of DNA. These are the components of purine which are naming 1 to 9. This 1 to 9. This N1 which is coming from the amino group aspartate, C2 coming from the tetrahydrofolate acid, and C6 which is coming from the respiratory carbon dioxide, and N1, N3 and N9 which is amide group of glutamine, and this group C4, C5 and C7 is glycine, and C8 is the methylene tetrahydrofolate. So we can see it, all these carbon atoms and the molecules which is coming from the different source. So that's why another important factor is, remember, to biosynthesis this purine nucleotide, this component must be present in your system, otherwise purine nucleotide cannot be synthesized. These purine nucleotides are synthesized by the most of the tissue. However, the major site of the liver, and this pathway operates in the cytoplasm, and the major pathway is denote the de novo synthesis. De novo means new synthesis is called the de novo, because the purine ring is synthesized from the different small components, and since the human being can synthesize the purine and pyrimidine bases, de novo synthesis. Actually, this big molecule coming from the different small molecule, that is called de novo, and they are said to be prototrophs. And this contribution of different atoms from different sources from the formation of purine ring, and during the de novo synthesis, purine ring is built up on a ribose 5-phosphate molecule. This purine nucleus is coming from the ribose 5-phosphate which is attached with the N9 position. Hence the nucleotide are the production of the de novo synthesis, and there are the 10 steps reaction of the de novo synthesis, and the enzyme catalyze this reaction are exactly existing in the multi-enzyme complex in the eukaryotic cell, and the arrangement increase in the efficiency of pathway. Biosynthesis is a multi-steps enzyme catalyzed process where the substrate are converted into a more complex product in the living organism. And the biosynthesis is a simplest compound is modified, converted into other compound and joined together to form a macromolecular substances. And this process often consists of the some metabolic pathway, that means there is some metabolic reaction, and the purine is built up upon the pre-existing ribose 5-phosphate which is coming from the pentose phosphate pathway, and this is the only pathway, pentose phosphate pathway or ribose phosphate pathway, by which glucose is converted, 6-member glucose is converted into the 5-member ribose 5-phosphate. Ribose 5-phosphate, it is an alternative path of the glycolysis. If the glycolysis is excess, then this pathway occurs, and this supply for the synthesis of ribose 5-phosphate, which is the part of the sugar part of the DNA. It is the, and that sugar part can initiate the synthesis of purine nucleus also. And the liver is the major site of the purine nucleotides. Other is the erythrocytes, polymorphonuclear leukocytes, brain cannot produce the purine. And there are the two pathways by which synthesis of purine nucleus, and one is the de novo synthesis, which is discussed in earlier, and biochemical pathway by the nucleotide are synthesized and to form the simple precursors. Another is the salvage pathway, that means to use to recover bases and nucleotide forming during the degradation of DNA and RNA. This is the another pathway. By de novo, it means totally new pathway by which small molecule combine together to form a new big molecule purine. But salvage pathway is another pathway, is purine coming from the diet or degradation of DNA or RNA. So it is the most schematic diagrammatic by which nucleotide is biosynthesized, not only the purine, in this case the de novo synthesis and the salvage pathway. In case of de novo synthesis, purine, pyrimidine, and salvage pathway, purine, pyrimidine. Purine is formation first of all IMP, then IMP to AMP, and AMP to ATP or GMP to GTP, and pyrimidine UAMP first, then DUMP, then DTTP, and UTP and CTP. This is the schematic representation of nucleotide synthesis and the de novo synthesis of purine. These are the discussed in the earlier. What is the structure of the purine nucleus? But all these components coming from the different sources, just like our god Devi Durga. They are their power is empowered by the different organs coming from the different goddess. The N1 coming from the group aspartate, C2 and C8 from the formate, and N3, N9 for amide group of glutamine, C4, C5, Cn from the glycine, C6, C6. So it can be seen that so many components is help for the synthesis of purine nucleus. That's why it is, remember it, all these components must be present inside in your body. Thus aspartate, formate, glycine and biochemical acts as a building blocker of the purine synthesis. Purine nucleus in this group, adenine and guanine, but adenine and guanine directly not produce, it is first produce inosine monophosphate. It added to inosine monophosphate. This is the schematic diagram of same as it is. The inosine monophosphate, this is the structure of inosine monophosphate, the purine nucleus with ribose 1-phosphate, that is called inosine monophosphate. And the steps are involved in the biosynthesis of adenine and guanine. First, ribose 5-phosphate and carbohydrate metabolism and the starting material for the purine biosynthesis, and the reaction with the ATP to form a ribosyl pyrophosphate, and glutamine for the synthesis of the amide nitrogen for PPR, phosphoribosyl pyrophosphate, to replace the phosphate and produce the ribose 5-phosphoribosyl, and catalyze phosphoribosyl glutamyl amidotransferase. The reaction is committed. The phosphoribosyl react with the glycine in the presence of ribosyl 5-phosphate, and the glycine in formyl tetrahydrofolate donates the formyl groups, and the products formed in the formyl glycinamide ribose 5-phosphate. These are the description of purine biosynthesis. Next, go to the schematic representation of our biosynthetic product. This is the steps for the purine biosynthesis. It is start from the PPR glutamine and PPR, then it is converted into the phosphoribosyl pyrophosphamide, then first glutamine plus PPR. glutamic acid and PPR plus 5-phosphoribosyl 1-amine In presence of glycine, they produce glycinamide ribonucleotide, and then tetrahydrofolate to tetrahydrofolate acid, formyl glycinamide ribosyl nucleotides, and glutamine is glutamic acid, is formation of formyl glycinamide ribose nucleotides. Then it converted step 5, the amide and amino imidazole ribonucleotides. Then it is converted to step 6, 5 amino 4 carboxylic amino imidazole ribonucleotides. Then the 6 aspartic acid which is added in this reaction, N-succinyl 5 amino imidazole carboxide nucleotides. Then the position steps number 8, fumaric acid is released, 5 amino imidazole 4 carboxamide imidazole. All these are given listed in the below. Then this compound, another step number 9, this is converted into the N-formyl amino imidazole 4 carboxamide ribonucleotides. Then step number 9, in the inosinic acid will be for or inosine monophosphate. Then aspartic acid to formation of phosphate adenylate acid or adenosine monophosphate AMP, first compound for purine synthesis. Then it is converted into deoxyribonucleic acid and ribonucleic acid. And this is the schematic representation structure of how to purine nucleotide form or inosine form from the ribose 5-phosphate to inosine monophosphate. See carefully. And this is the pathway, complete pathway of this, where the inosine monophosphate coming from the ribose 5-phosphate. The synthesis of GMP, AMP, GMP from IMP. Actually, first precursor molecule is the IMP, which is coming from the first 10 steps of reaction from the ribose 5-phosphate. Inosine monophosphate, it is the immediate precursors for the formation of AMP and GMP. And aspartate condensation with the IMP, which presence of GTP to produce adenyl succinate, which is on cleavage to formation of AMP. This IMP undergo the NADP dependent dehydrogenase to form a xanthine monophosphate. And glutamine, which is transfer the amide nitrogen xanthine monophosphate to xanthine and to produce GMP. 6 marcopurine is the inhibitor of AMP and GMP, and 6 marcopurine, that's why used as one of the most important anti-cancer drug for the inhibit the synthesis of AMP and GMP in case of viral DNA or in case of cancer cell. It acts as the enzyme adenyl succinase, AMP pathway or IMP dehydrogenase. So this is the pathway of AMP and GMP, how it will be produced. The AMP in presence of aspartic acid as adenyl succinate synthase, it converted to adenyl succinate. This adenyl succinate is converted to AMP, that means adenosine monophosphate. Then AMP again phosphorylation adenosine diphosphate, and then again phosphorylation, it will be formation of ATP. This IMP, another pathway, it is a NADH dependent pathway. In this case, NAD is converted to NADH and is xanthine monophosphate, xanthosine monophosphate, and xanthosine monophosphate to GMP synthesis is converted to GMP to phosphorylation GDP, and then guanosine monophosphate, then GTP. This is the pathway by AMP to formation of AMP and GMP, which is the another precursors for the synthesis of base for the nucleotide. And this is the structural formula of inosine monophosphate to adenosine monophosphate or guanosine monophosphate. Another is the salvage pathway, that means in this case that AMP or GMP or IMP, which is produced directly from the foods or from the degradation of DNA RNA. This adenine, which is adenine phosphoribosyl transferase, which is converted to AMP. Guanine, which is present in the or degraded product, hypoxanthine guanine phosphoribosyl transferase, it converted to GMP, and hypoxanthine is converted into a hypoxanthine guanine transferase IMP. This is the totally different reverse pathway by which AMP GMP is produced, not directly synthesized. It is the degraded product of adenine, guanine, or DNA, RNA, or from the diet. And conversion of ribonucleotide to deoxyribonucleic acid. This is the base which are used for the ribonucleic acid, but how it will be converted to deoxyribonucleic acid? Actually, the starting material ribose 5-phosphate, which is the important factors. The synthesis of purine and pyrimidine deoxyribonucleotide occurs from the ribonucleotide by the reduction of at the C2 of ribonucleotide, which is earlier discussed in the very briefly discussed in case of nucleic acids and structure of the DNA. In that case, the oxygen is divided in the position number 2, and this is called the deoxyribose. This deoxyribose is the starting material for the synthesis of deoxyribonucleotides, and this reaction is catalyzed by the multi subunit 2 B1 B2 subunit enzymes ribonucleotide reductase. This is the pathway by which the ribonucleotide diphosphate ADP, GDP, CDP, or UDP, which is converted to ribonucleoside diphosphate ADP, GDP, CDP, and UDP, and ribonucleotide reductase in the presence of thioredoxin 2-SH reduced to thioredoxin SH, which is again converted into a thioredoxin for the next steps of reaction by the thioredoxin reductase. Here ribonucleotide reductase that converted into a deoxyribonucleosides to deoxyribonucleoside triphosphate, and thioredoxidation. And this is the pathway by which the ribonucleoside triphosphate is converted into a ribonucleoside diphosphate. Ribonucleoside diphosphate, first the reaction in case of it is the deoxyribose, that means the component of DNA, but here in the right side ribonucleotide reductase, the oxygen is usually removed, that is called the ribonucleotide diphosphate. All are component for the RNA or ribonucleosides, and supply the reducing equivalent the enzymes ribonucleotide reductase. It is provide the hydrogen atom needed for the reaction for the sulfur hydroxyl group. The reduction equivalent enters in the supplied by the thioredoxin and monomeric protein to cysteine residue. NADPH dependent thioredoxin reductase converted to oxidized thioredoxin to reduce form of it catalyze again and again for the next reaction, and thioredoxin are several protein cofactor in the enzymatic reaction. The regulation of deoxyribonucleotide synthesis. The regulation, this is very important. Every reaction, every pathway can be regulated, otherwise it will be a problem in case of not only your metabolic reaction but also in case of your life also. There must be regulation, otherwise it will be not under control, not control. The deoxyribonucleotide are the mostly requirement for the synthesis of DNA, and the enzymes ribonucleotide reductase maintain the adequate supply of the deoxyribonucleotides. And ribonucleotide reductase, it is a complex enzyme which is complex multiple site, active site or allosteric site that control the formation of deoxyribonucleotides. For the inhibitors of purine biosynthesis, basically the tetrahydrofolate, the folic acid, which is essential for the synthesis of purine nucleotides. Sulfonamides are the structural analog of aminobenzoic acid or PABA, and the sulfur drug can inhibit the synthesis of folic acid by microorganisms. That's why the PABA is the sulfonamides is the most important antibacterial agent for the synthesis of purine nucleus, and that organism or bacteria can be bacterial growth can be inhibited. The indirectly they reduce the synthesis of purine and nucleic acid, DNA and RNA. The structural analog of folic acid and method, just like as for example methotrexate, which is used for the anti-cancer drugs, and azathioprine, which diazocytyl L-serine is a glutamine antagonist, inhibit the reaction involved the glutamine, and other synthetic nucleotides analogs used for the anti-cancer agent for 6-thioguanine to as a thioguanine. This is the inhibitors. For PPR, the phosphoribosyl pyrophosphate, it is a donor of ribosyl pyrophosphate and the steps 0-step reaction first or pre-step reaction ribose 5-phosphate to form a de novo synthesis, and this reaction ribose 5-phosphate ATP ADP plus 5 phosphoribosyl pyrophosphate or PRPP. The structure of PRPP, the purine ring is later it is assembled on the ribose 5-phosphate, and PPR also used for the synthesis of pyrimidine nucleus also. So PRPP is the most important precursors for the synthesis of purine nucleus, not only the DNA but also the RNA also. Nucleotide coenzymes that also have the salvage pathway, hence the synthesis of PRPP, it is the considered as steps for the de novo synthesis of purine nucleotides. It is called the preliminary or preparative steps. Formation of AMP, this is the structure. The salvage pathway, which is already discussed. From this pathway, adenine persistence ADP AMP plus PPI, guanine by PPA, GTH enzyme GMP plus PPA, hypoxanthine PRPP, and the enzymes involve IMP and PPI. Then the regulation of purine biosynthesis. The committed steps to de novo synthesis is reaction catalyzed by the amide transferase step 1. It is inhibited by the AMP and GMP. There are allostatic modifiers. Bindings of AMP and GMP on the coenzymes convert to monomeric active form to dimeric ingestion. And in the GMP and AMP can bind with the same enzymes molecule at different sites at synergistically inhibit the enzymatic action. Ultimately the steps will be inhibited. Both AMP and GMP inhibited in the own formation of feedback inhibition by adenosine succinate synthase and IMP dehydrogenase. And the formation of another step 5, formation of AMP to IMP required for the GTP, and similarly formation of GMP required to ATP. Hence the GTP and ATP are made the available for the sufficient quantity. The availability of PPR, it is another important regulatory factors, because PRPP, which is the main precursor for the synthesis of purine nucleotides, the activity of PR synthesis is regulated by the negative modifiers purine and pyrimidine nucleotides. So though we can see that how the purine nucleotide synthesis from the PRPP and ultimately inosine monophosphate, then inosine AMP, GMP, AMP to ATP and GTP. That is the base of purine type of nucleotides, when purine bases which is attached with the sugar along with the phosphate, their formation of purine nucleotides. So I think you can understand very easily, and thank you listening for your patience, and thank you all.